brief history of microbiology. It is very important before we enter into the subject of microbiology. What had been done in the olden days at its inception stage. First of all, Leeuwenhoek in 1632 to 1732. He was a Dutch scientist and he prepared lens of the magnification of 300 times. And this building up of lens had helped him see the bacteria, which are some are rods, bacilli, some protozoans, eggs, yeast, and RBC, spermatozoa were the first cells which were sighted under microscope sort of thing. Thereafter, Edward Jenner in 1796. What he did? He just prepared the process of immunization, that is vaccination, by using the material from the infected part of the animal and just inoculating in the healthy ones. Thereby some antibodies they got developed, and this was how the first vaccine was developed. These days there are several ways of developing the vaccines. Among them one of them is inoculating attenuated organisms so that they may not cause the disease in the person which is getting immunized. Then after Edward Jenner, a very important and noted scientist or microbiologist came into the scenario. He was Louis Pasteur. You all might have heard about Louis Pasteur. He lived during 1822 to 1895. What he did, he just find out the process of fermentation, and those fermentations were microbes associated, and he formulated a microbial theory of fermentation. Among the fermentation procedures, one of them is alcoholic fermentation. In this, the substrate which was used by Pasteur was grape juice, and the microorganisms were acid resistant yeast cells, and from this fermentation he got alcohol. Then we have lactic acid fermentation. You all know what is lactic acid fermentation. The substrate is milk and the microbes used are lactobacilli. This is called as acid sensitive bacteria. These are called as acid sensitive bacteria. Then most important procedure Louis Pasteur developed was pasteurization of milk. In pasteurization, the milk was first heated to 62 degrees centigrade for 32 minutes. Thereafter, for 30 seconds, the milk was subjected to heat at 72 degrees centigrade. Thereafter, milk subjected to instant chilling. And this is the, whenever you see a label of pasteurized milk, this is the procedure how the milk is pasteurized. Then another important discovery he made was formation of anthrax and rabies vaccine. Okay. Then came Robert Koch. He put forth germ theory of disease, and he discounted the earlier existing theory of spontaneous generation. In this theory, germ theory of disease, what he postulated, organisms found in lesions of infected persons but were absent in healthy ones. Means he could discover some microbes in the lesions of the diseased person. And those microbes were absent in the tissue of healthier ones. Then the microbes which existed in the diseased person, they could be isolated and a pure culture can be formed out of them. If that pure culture is inoculated in healthier ones, the healthier one used to catch the infection. Okay? And the healthier one who had caught the infection, even from that one we can isolate re-isolate the suspected animals. Okay? Then came Sark and Savy. They invented a very important vaccine called as polio vaccine, which used to prevent the person from suffering from polio. Then Fleming, Chain and Florey, they made an epoch-making discovery by finding out penicillin in 1945. It played an important role during the World Wars for preventing the soldiers from infection caused due to wounds. Waxman thereafter discovered another antibiotic termed as Streptomycin. Then Beadle, Totem and Liddell, for the first time, they discussed or described the microbial genetics. Nathan, Smith and Arthur, they discovered restriction enzymes. The restriction enzymes are cleaving enzymes which cut the nucleic acid at specific sites. These days, these restriction enzymes or endonucleases, they are widely used in biotechnology or recombinant DNA technology, where you require to cleave the nucleic acid and remove the segment which is cleaved and inserting the sequence of nucleic acid containing desirable gene. Then Mullis and Smith, they invented a technology which is called as PCR, polymerase chain reaction technology. What it does, even if we are having a few fragments of nucleic acid, they can be amplified and majorly used in DNA fingerprinting. Then Joseph Lister, he was the first who developed the antiseptic surgery. Fleming, he discovered lysozyme. You all know where lysozyme exists in our body. Where does it exist? Lysozyme is present in the saliva. That's why whenever we inadvertently get pricked our finger, we intend to place it in our mouth, because the lysozyme present in the saliva, it lyses the bacteria on the wound. Then Stanley, Cohen and Herbert Boyer, they developed a technique which is called as recombinant DNA technology. You might have heard about BT cotton and all that. The BT cotton is developed from recombinant DNA technology. That means recombination of DNA means that combination of genes which earlier existed has been tweaked, and new genes are inserted in the genome and get it expressed in form of proteins. Chamberlain first of all discovered bacterial filter. This was an important invention in the sense that it has led to the discovery of viruses. Without bacterial filter, virus could not have been discovered. Ivanovsky for the first time in 1882 discovered virus, and the first virus discovered was tobacco mosaic virus. Stanley in 1935 first of all crystallizing the viruses. This crystallization of viruses is very important because this denotes that the viruses are non-living, because only non-living thing can be crystallized, not a living thing. Toad and Deagle first of all discovered bacteriophages. What are bacteriophages? Bacteriophages are the viruses which infects the bacteria, and they complete their cycle in bacteria by directing the bacterial genome to form the viral components. Hershey in 1952 stated that it is not the entire virus, but the nucleic acid of the virus, which contains the genetic information, only enters the host, not the entire virus. And in the last, Tamming and Barty, they discovered RNA-dependent DNA synthesis. It is done by reverse transcriptase enzyme. As I have told you when I described AIDS virus, that reverse transcriptase helps virus in converting their RNA into double-stranded DNA, so that it may get integrated into host genome